My name is Cassandra Ortega, and I'm going to discuss the role of p53 in cell cycle regulation. This captures the discussion of p53's function, research findings, and the consequences of mutation. Role of p53. It's a crucial tumor suppressor and plays a key role in maintaining genomic stability by regulating the cell cycle. Main functions: cell cycle arrest. p53 holds the cell cycle at specific checkpoints to allow time for DNA repair. When DNA damage is detected, p53 becomes activated and induces the expression of p21. A cyclin-dependent kinase inhibitor. p21 binds to and inhibits CDK cyclin complexes, preventing the progression through the cell. DNA repair. p53 facilitates the transcription of genes involved in DNA repair pathways, giving the cell an opportunity to correct damage before dividing. Apoptosis. If the DNA damage is irreparable, p53 initiates apoptosis to eliminate potential harmful cells. It does so by upregulating pro-apoptotic genes such as PAX, PUMA, and NOXA. Senescence. p53 can induce a state of permanent growth arrest called cellular senescence in response to prolonged stress or damage. Regulating genomic stability. p53 ensures that cells with damaged DNA do not proliferate, reducing the risk of mutations and cancer development. By preventing the proliferation of damaged cells, p53 acts as a defense mechanism against cancer formation. This analysis is based on the article titled "p53 promotes cell survival due to the reversibility of its cell cycle checkpoints." Objective: the experiment aimed to investigate the role of p53 in regulating cell survival and cell cycle checkpoints after DNA damage. Specifically explored how p53 contributes to reversible cell cycle arrest, DNA repair, and prevention of apoptosis under different levels and durations of DNA damage. On our right-hand side is Figure 5, specifically from the article. As I mentioned, it demonstrates the recovery of p53 expressing cells after transient DNA damage and their ability to resume the cell cycle, contrasting the irreversibility damage in p53 null cells. Cells were exposed to deoxyriboxine for 6 hours, followed by drug removal and recovery. DNA content and markers of DNA damage, YH2AX, were monitored. And results: p53 expressing cells recovered and resumed cycling with reduced DNA damage. p53 ablated cells remained arrested or under apoptosis, indicating by persistent YH2AX staining and hypodiploid DNA content. So p53 is crucial for transient arrest and recovery, enabling survival and genomic stability. The mutant TP53R248Q gene exhibits a gain-of-function mutation commonly associated with high-grade serous ovarian carcinoma. Instead of acting as a tumor suppressor like wild-type p53, this mutant form contributes to oncogenesis by enhancing AKT signaling, a pathway that promotes cell proliferation, survival, and drug resistance. On the right-hand side, it's figure 1 from the article. It shows how the TP53R248Q mutation increases the activation of the AKT pathway in cancer cells, particularly in a high-grade serous ovarian cancer cell line, OVCAR3. So scientists observed a protein called PAKT, which signals cells to grow and divide. They compared cells with TP53 R248Q mutation to normal cells and added a growth signal, EGF, to see how PAKT levels changed. Cells with this mutation already had higher PAKT levels, even without extra signals. When the growth signal EGF was added, the PAKT levels got even higher in these mutation cells compared to normal ones. The p53 R248Q mutant makes the growth pathway hyperactive, causing cells to divide so much. And this helps explain why cancer cells with this mutation grows uncontrollably.